Yay. Hi, everyone. Welcome back to the 30 by 30 track. I have the pleasure of welcoming 7 experts on species to speak in this session. So monitoring technologies are collecting information about global biodiversity at a rate and resolution that is like unfathomable 10 years ago, a decade ago. And we're gonna hear how this data is informing decisions and leading action to protect, manage, and restore nature and biodiversity. So we have a lot of talks in this session. We're gonna aim to have some time left at the end of this session for Q&A. But if we don't, we have a facilitated discussion at the end of this, in our third 30 by 30 session at the end of the day, immediately following this session in Discovery B. So our first 2 speakers are Jorge Ahumada from Wildlife Insights and Lindsey Rich from California Department of Fish and Wildlife. Jorge, over to you.
Thank you. Thank you. Thank you, Tanya, for the invitation to the event. I'm really honored to share this talk with Lindsey from the California Department of Fish and Wildlife, and we'll be talking about using camera traps to measure species outcomes from for 30 by 30 and beyond. So, about 189 countries have agreed to the Kunming-Montreal Agreement, the CBD Agreement. We heard about this earlier in the session on habitats. I highlighted here the goal A and target 2, but highlighted especially what has to do with species. So these are very ambitious goals. Stop the extinction of threatened species, reduce extinction rates by 10-fold, ensure that the abundance of wild species increase to quote-unquote healthy levels, and then target 2, which is very important, recover 30% of areas that are degraded in the terrestrial realm, inland water, and coastal are effectively restored by 2030. So, this is all very good and ambitious. How are we supposed to deliver on these goals? And there's been several indicators that have been proposed. All these indicators, I'm not going to go into detail, but all these indicators require data on species on the ground. So camera traps are really also called trail cameras or trail cams. They're really one way to do this at scale. And there's other sensors that we'll be talking in this session, but I'm going to focus on trail cams. And they have several advantages listed here. One of the greatest things is that the data is verifiable. Allows for many different species to be sampled using the same methodology. And they sample an important subset of terrestrial mammals and birds, which account for a large portion of the terrestrial wildlife biomass. So in an attempt to give visibility to the thousands of camera trap projects around the world, we know several of us, the partners listed down here, and Google, we've created Wildlife Insights. It's an AI and cloud-based platform to accelerate the processing of camera trap data, removing most of the main pain points experienced by users, empower users to manage and analyze their own data to improve wildlife and conservation outcomes, and also provide a vehicle for data sharing and collaboration. And since becoming public in 2021, Wildlife Insights has received more than 1,300 camera trapping projects coming from 95 countries and resulting in more than 140 million images. So because of its global nature, Wildlife Insights can be used to answer questions about the status of terrestrial mammals and bird biodiversity at various scales. An important user segment of Wildlife Insights includes protected area stakeholders, including government agencies, protected area managers, scientists. And so we estimate that around 883 protected areas larger than 100 square kilometers have used camera traps to monitor wildlife. And other users of Wildlife Insights include land trusts, NGOs, researchers, and others, and governments, of course. This kind of nature of the data allows us to ask questions like this. Very simple question: do trail cameras detect more species in protected areas than outside of it? And these questions are difficult to answer with normal data because of the nature of the data. But here we have a camera trap that is sitting in space somewhere, sampling for a certain amount of time doing it. So when we crossed over the World Database of Protected Areas with the Wildlife Insights dataset, this allows us to identify where camera traps were deployed and whether they were inside or outside of protected areas. About 51,000 camera traps, which actually is a subset of the whole dataset. I'll explain in a minute. And of which 19K were in protected areas, about 32K in outside of protected areas. We kind of narrowed the cameras that were deployed between 2003 and now, so 2023. And we only also use cameras that were functional between 20 days and 100 days. And we also narrowed down the species to birds and mammals, because there's other species like reptiles and small number of amphibians that can get into the data. We didn't want that to be contaminated. We used a very, very simple Poisson regression model to measure the effect of being in a protected area on the number of species detected in a camera trap. So this number can be influenced by many different things aside from the protection status, like effort, latitude, longitude, and all continuous covariates were standardized to a normal distribution with mean 0 and standard deviation 1. So this is a very, very simple model that can affect species richness, but it ignores other important effects such as spatial autocorrelation. And for now, we ignore these complications. So the results should be considered very exploratory, very, very preliminary. So what did we find? First, there's a substantial variation in number of species captured in camera traps, ranging from 1 to 72. The mean is about 4 to 5 species. Now, when we look at the effects, we see that here the effects plotted by protection, longitude, latitude, and effort. We see that being in a protected area that is highlighted there results in about a 10% increase in the number of species detected. This is after correcting for all the other ones. This other graph shows the predicted richness. Each point here is a camera trap recording a certain number of species. And you can see that there's a subtle but important difference in richness between the two. Finally, and one thing that I found very interesting is when you start looking at this data by country, then you see substantial differences. So, for example, the effect is very pronounced in Bolivia, similar to the mean in Kenya and Peru, but doesn't look like in Australia you can detect this, and it might be because of differences in richness at the scale of these countries or other factor. So now I want to hand it over to Lindsey to look at another example of how camera trap data and wildlife insights can be used to make decisions at the state level. Thank you.
Thanks. So CDFW formed a collaboration with Wildlife Insights about 4 years ago, CDFW's California Department of Fish and Wildlife. So I'm just going to provide an additional example about how the data can be used and the power of aggregating all of this information in one centralized location. So the goal of our study was to use existing camera trap data to identify areas of conservation concern due to cannabis cultivation in the predominantly forested areas of Northern California and the Sierra Nevada. The first step that we took was to aggregate all of the camera trap data that we could find from 2019 to 2021. And this was really easy because it was in Wildlife Insights. We found data from 10 different projects, including 1,186 cameras. And these cameras provided information on 30 different mammal species, some of which are shown here. Then we use this camera data in combination with spatial covariates to estimate the likelihood that each of these animal species occupied a place of interest, and then extrapolated the model results to our entire study area at a 1 square kilometer scale to generate detailed distribution maps for each of the species, like the example shown here where you have mountain lion on the left and the American badger on the right. Our second step was estimating the risk of trespass cannabis cultivation as well as unlicensed cannabis cultivation on private lands. And these are the maps, the two maps on the left. To do this, we built Maxent models using the locations of known cultivation sites and an array of spatial covariates. For the licensed data, we used the locations of mixed light and outdoor cultivation licenses. And then the final step was to identify spatial overlap. To do this, we used a bivariate mapping approach that classifies each layer into 3 quantile bins to emphasize areas of high versus low overlap. So the areas that are purple show large amounts of overlap. And so for here, the American badger is on the left, and then species of conservation concern richness is on the right. And tools like these are extremely useful for the department because now we can target our management actions in places of greatest concern. We also have grant programs, and so we can allocate grants in places where we're going to get the biggest benefit to biodiversity and conservation in the state. If you're interested in learning more, there's a link to the paper that's at the bottom of the slide. And then just to bring it back around to Wildlife Insights, California Department of Fish and Wildlife is up to over 45 million images from 13,500 camera deployments and 46 different projects. This was absolutely not possible without being able to use Wildlife Insights, because these projects range from a statewide bobcat effort to drought resiliency to tracking the spread of nutria and invasive species in California. Before, all of this information was in disparate locations, and now it's together. We can process the information more efficiently, and I'm just really excited to see all of the additional applications that the department will be able to use to help us make science-based data decisions. And at the bottom of the slide, there's a QR code for our field-based protocols. We also have a statewide biodiversity effort that's part of California's 30 by 30 effort. So if you're interested in checking those out, please do. And if you have ideas on how the amazing technologies that you're developing could be integrated with our field-based sensors, please reach out and email me, because I'd be happy to talk. Thanks.
Thank you so much, Lindsey and Jorge. Next up, we have Bourhan, who's the CEO of Rainforest Connection, speaking with Lilian from the Jane Goodall Institute, VP of Conservation Science. Welcome.
Yeah, thanks, Tanya. Hello, everyone. So my name is Bourhan. I'm the CEO of a nonprofit called Rainforest Connection. We are a project that's supported and funded by Google.org. I'm here to talk to you about the power of sound to analyze and discover species. What I'm about to play is a small video that is combined with using convolutional neural networks, AI, to discover species in just a 30-second clip. What would be great for you to do is to try to see if you can actually pick out these species yourself with just hearing it, as you see the AI detecting the particular species that are popping up. This is a 30-second clip from a project that we have in Panama. Can we roll the video, please? So you can see here the AI is essentially detecting several species, birds, howler monkeys, parrot, and is just doing this at an extreme precision. If this was a little bit louder, it'd be very difficult to pick out amphibians from birds, from insects, and so on. And guess what? During this whole time, there's actually a chainsaw. If we can turn it up a little bit more, there's actually a chainsaw that's running the entire time. You could only hear it because we turned everything else down, but that chainsaw was running from the beginning. And our human ear actually couldn't hear it, but with an AI, with the power of AI, we can actually hear all these things. So not only we can decipher all this stuff that you see in just 30 seconds of audio, we can actually hear and detect illegal activities as well, like chainsaws, which are indicative of illegal logging. So how do we do this, right? We use these inexpensive recorders that we mount on top of the tree canopies. They're called the Guardians. And they are essentially just glorified sound recorders that have solar power, batteries, all that kind of good stuff. And they're designed to record the soundscape 24/7. And they can either run machine learning on the device itself or, even better, upload that to the cloud. And that's when we use AI and machine learning to actually analyze all this audio. So we can pick out very specific species, like you saw earlier, down to the individual type, sometimes even down to the song itself or the call itself. And we can also pick out really bad stuff, right? We can pick out chainsaws, indication of illegal logging. We're losing over 12 million acres of rainforest every single year, threatening over 1 million species with extinction. It's a really big problem. We can pick out gunshots, which are indicative of poaching. And we can pick out trucks or any kind of forms that people and humans are using to bring wood and all sorts of things in and out of the forest. But most importantly, and what Dr. Lilian would talk about later, is our capability of using that for biodiversity monitoring. One of the most profound ways of being able to study biodiversity is just to listen. Listening is one of the most important tools at really being able to pick out species. And if you can tell the species and the species type, you can start looking at richness and occupancy, and you can start looking at counting the species and eventually be able to get to biodiversity health, which is very important. Now, these tools are very advanced, sometimes very complicated, but there has to be a way where we can bring these tools down to the people that are on the front lines, that are actually the ones who use this information and make a difference. All these local communities, all these local scientists in many different countries around the world, they're the ones who need to use these tools. And very similar to what Jorge has and the amazing tool that's Wildlife Insights, that brings this capability to a no-code approach for scientists that are interested in the science and less interested in the technology. We've designed a tool that does the same thing for sound, and that's called Arbimon. So, backtracking a little bit, Ecoacoustics, which is the science that's been there for decades and decades, that is using sound to study biodiversity. It's a very non-invasive, a very easy-to-use science. It covers a very large area, the ability to record soundscapes. You can do it over long time frames. It's a very good way. Lots of indicator species are actually vocal, birds, for example, and amphibians. You can survey multiple species simultaneously because you're recording the entire soundscape. So ecoacoustics, which is what I've been talking about for the last few minutes, is the science of using sound to understand biodiversity. And the most important thing, and I think that's something that we often miss, is the ability for ecoacoustics or for sound to be a permanent record of the soundscape that you're recording at this point in time. And the reason why that's important is because we don't know right now what we're going to do with this soundscape later in the future. Technology advances, machine learning models advance. We have the capability to run over this data over and over again to be able to pick out more interesting insights out of it. And the reality on the ground is, with climate change and everything else that's going on, we're never going to hear the same soundscape ever again. So this is a moment, we're capturing a moment in history with recording soundscape and storing them forever, because we're never going to get to hear it again. The soundscape in Brazil is not going to be the same now that it's going to be 5 years from now. So keeping that record is very important, and ecoacoustics allows you to do that. So Arbimon and Lilian will spend time, and he has some interesting stuff about an actual project on the ground that's using Arbimon. But Arbimon is an ecoacoustic, no-code approach online platform that allows you to upload unlimited amounts of data completely for free. Many thanks to Google.org for the support on that to allow that to be possible. But the important thing about being able to detect species through sound is then we can start doing all kinds of things, like looking at species richness, looking at the most suitable areas where species should and will thrive, and many other things that my colleague will talk about later. But you can see this is an example of the insights that comes out of Arbimon. And this is all automated. As soon as the CNN is running over the data, the machine learning model that we talked about, you can see all that information starts popping up on a species level, right? And so what does that mean? Well, Arbimon now is the largest ecoacoustic platform in the world. It's used by over 5,000 scientists in 124, 123 different countries around the world. There's over 1.3 billion analyses that have been done on Arbimon to date. That includes machine learning models, includes other techniques of filtering through the data. Over 135 million recordings have been uploaded to date. If you were to put that in other terms, it's almost 220 years of continuous soundscape data, soundscape audio. Probably by far the largest soundscape collection in the world, perhaps even in history, that's been uploaded from all these users around the world using the techniques that are on Arbimon. What would that allow us to do? So all these little mini projects that are happening, or some of them are big projects, allows us to do global level reporting, being able to understand species migration, species richness, being able to understand biodiversity hotspots, and hopefully direct funding and direct projects towards that. That's all possible with the capability of collecting large amounts of data and being able to analyze that. So that's all I have to say, but there's more exciting stuff with what Lilian has with a project on the ground. Dr. Lilian Pintea from Jane Goodall Institute. Dr. Lilian.
Thank you so much. Thank you. So in 1960, when Dr. Jane Goodall arrived in Gombe, the world changed. She opened this new window into chimpanzees, into the world of other animals, and into ourselves. So today, the Jane Goodall Institute works across the entire chimpanzee range in Africa, and we have our roots and shoots groups around 67 countries around the globe. And we are continuing the long-term research which Jane started. Now Gombe is the longest ongoing great ape research site in the world. We are combining Jane's approach with the latest innovative technologies from DNA to satellites. So you can imagine how excited we are when we had this opportunity to partner with Rainforest Connection to start deploying the bioacoustic devices so we can start learning how these new technologies can be added to the toolkit of other toolbox of technologies we're using and how it can help our long-term research and conservation efforts. Now, Bernie Krause, the famous musician and sound ecologist, already recorded some soundscapes of Gombe back in the 1970s and also 1990s. This is truly remarkable because speaking about this historical library of sound, Gombe back then looked very, very different, as you can see from the satellite imagery, with less forest inside the park, but with much more forest and woodland and better connected with forest outside the park, which has been declining since then. So at the Jane Goodall Institute, we are really excited to start using our best GIS layers, mostly derived from Maxar satellite imagery, to meter vegetation layer, house density, footpath, elevation, and so on, to capture this gradient both ecologically and in terms of human disturbance. So we selected our 100 sites to deploy these audio devices. This was also combined with building our capacity of our research team at the Gombe Stream Research Center, of our colleagues in Tanzania National Parks, ecologists from Tanzania Wildlife Research Institute, and other partners to deploy, manage, upload these devices. Now, one problem which we had to solve is that the baboons destroy everything like this in the forest. And we actually had a special line in our google.org proposal, money to be spent on baboon-proof cages. Well, it didn't work out. And I'm so amazed by innovation of our local team, who figured out that just applying this disgusting grease on devices looks not only disgusting for us, but baboons also don't want to have anything with it. So from 100 devices, we only lost 4. Now, we're still in the middle of analyzing the data, but I'm so excited to share with you some truly incredible insights already. First of all, more than almost 90% of the species detected were birds. This is really important because we knew that there are birds in Gombe, but this provides us really an important tool to understand better this biodiversity. Now, from the mammals side, this is incredible. The first 2 weeks of deployment, we were able to find a new bush baby. And this Thomas dwarf galago is well distributed in West Africa, but it's the first time being present at Gombe and also in Tanzania, which will result on substantially extending the species range. And isn't it incredible that Gombe probably is the most visited site by primatologists in the last 60 years, and in the first 2 weeks, we were able to make this discovery. Now, even the long-term studied primates like baboons, we are able already to get some interesting insights. For example, all the long-term research behavioral follow-ups which we have is during the day. But baboons, of course, have very interesting and dynamic lives at night, as you could see here from the detection frequency. And our primatologists studying baboons are very excited to start looking at the bioacoustic data to get those additional insights into baboons' lives. Now look at the map. You see that the spatial distribution and density of these activities is also quite remarkable and interesting. And this is exciting for our disease experts who are modeling the potential disease transmission hotspots using One Health approach between people, livestock, animals, and the changes in the environment. This, for example, in red are the baboons reported by the village forest monitors using smartphones. So this is truly remarkable because the disease is one of the major threats to chimpanzees and other wildlife in Gombe. There are 3 chimpanzee communities. The northern Mitumba, the middle one, Kasakela, are the ones which were studied and they habituated. But the community in the south, Buavi, it's not habituated, and we know that it has been steady declining over the years because of disease, and including the SIV, the major one, which is the virus contributing or jumped to the humans to became HIV. And it's remarkable that we're able to detect 2 or 1 maybe vocalizations from chimpanzees in the south, which indicates to us that still there is some individuals left in that community. So we're looking forward to start better understand what's happening to that community if it will be extinct or will be able to bounce back. What I'm really excited about is now connecting this dynamic, continuous data source from bioacoustics with huge library of continuous remote sensing data in Earth Engine. Back in 2012, I remember the Google team with us presented the first chimpanzee habitat suitability model run in Earth Engine at the Rio conference. And back then, we have to use nesting locations. And most of the time, we cannot afford to go and do surveys in wet season and a dry season. So you're kind of combining it all together. Well, now we have the means to truly develop the seasonally dynamic species distribution models because the chimpanzee behavior and ranging patterns in the dry season and wet season are remarkably different. And when I'm thinking about and looking at that network of distribution which Rainforest Connection is maintaining, it's truly inspiring because we can now model not only seasonal distribution, but also across maybe the entire chimpanzee range in Africa. And I still hope that we'll be able to compare the soundscapes of Gombe back in the 1970s with now. So to be more discovered, and we'll share with you later. If you're interested in this work, please approach. I'll be very excited to talk with you about collaborations. Thank you.
Thank you so much. So next up, we have Karis Tennyson from Spatial Informatics Group. A couple eDNA talks up next.
Hi, thank you, everybody. Before I jump in, I need the... Before I jump in, I need this. And second, just to wake us up, how many of you have had a piece of chocolate or some chocolate this week? Okay. So my talk is a bit about how can we assess the impact of investments on more sustainable, more conservation-friendly practices for providing us with some of these foods that we love, that we know do have an impact, driving some of the conservation issues we're hearing, the whole panel is talking about all day today. So you've already seen we've got a biodiversity challenge. At the same time, we're seeing increasing commitments to protect biodiversity. How do we measure the efficacy of these commitments, these interventions, how we're spending our money to protect, conserve species? So I'm with Spatial Informatics Group, we're an implementing partner of the Servir Amazonia Hub, which is a joint initiative with NASA and USAID. And we're partnering with another group sponsored by USAID to kind of investigate this question of how can we use remote sensing, but also some in situ data to assess the effectiveness of sustainable agriculture practices or restoration practices in improving conservation. USAID in Brazil recognizes that some of the key drivers of forest loss impacting species around the world, but especially in Brazil, is driven by economies. Communities need a sustainable economy. So their mission is engaging with the private sector, and they're one of the co-founders of the Amazon Biodiversity Fund. And through the Amazon Biodiversity Fund, they sponsor, either through low-interest loans or grants, a number of, I skipped the slide before I thought I did. Anyways, they sponsor a number of deals for farmers, for companies in the area at the interface of, where you have agriculture occurring, where these communities are living, and at the interface of the Amazon, to do sustainable agriculture. And one question they had is, how do we measure, is our money making a difference? So we worked with CIAT and a team with Servir again to develop some methods to answer that question. And for a bit of background, our pilot study that we applied 2 techniques to was assessing how species or conservation, quantifying species improvements, the improvement of conservation at a 15-year-old site where degraded pastures had been converted to shade-grown cocoa with the diversity of plants kind of interspersed within them. So we started with remote sensing. So we looked at some of the classic measures you've probably already heard about this morning. Are we seeing changes in habitat connectivity? Are we seeing changes in tree cover gain? If we expect, restoration to be happening, tree cover being planted above cocoa, are we seeing that with the imagery? Are we seeing unintended consequences? Is there forest degradation occurring at the periphery? Do we see forest loss happening? Remote sensing is amazing in giving us wall-to-wall information. So we calculated a lot of these metrics. But the next question is, forest tree cover can look very different when you look under the canopy. So how do we measure what's happening under the canopy? We considered acoustics, and I love hearing more about all of this research. And ultimately, we landed on doing an inventory, in situ inventory, using eDNA. When I went to my Airbnb the other day, I passed by 23andMe. Have you guys heard of that? You stick the Q-tip in your mouth, you put transfer some of your DNA to the Q-tip, send it to the lab, and they run it through their database, and they provide information on your genetic heritage. We're doing the same thing here, where we're collecting samples from the environment where we know animals and plants have left traces of their genetic material, so spit, butterfly wing scales, skin. We collect that. In this case, we're collecting it in soil, preserving it, sending it to a lab, and they run that soil sample through their library. They do genetic sequencing, run it through the library, and they're able to return back to us information about all the species that have been present in our site. So how are we using this? For these deals, the farmers that are getting, low interest loans or grants, we know where they're located. We know what their intervention types are. We also know about the landscape. We know where the adjacent, more or less intact forests are, and we know where those business as usual scenarios are. So we send our team to go work with the farmers and train them how to collect soil samples. We identify the sites and they grab samples from any of their different intervention types. They grab samples from adjacent forests, and then they grab samples from a business as usual, their neighbor, or sometimes, they apply the interventions to some of the fields in their farm, so they just sample business as usual on their farm. For the pilot, we did this on data for a site that had been established for 15 years and got really interesting results. For assessing the impact of interventions through the Amazon Biodiversity Fund, we have the ability to go out there and take these measurements every year. So what we expect to see, and we saw this with the data that we fast forwarded to on our pilot looking at 15-year-old cocoa, for the sites that we're starting to monitor now as part of the Amazon Biodiversity Fund, this is what we expect to see. So in this plot, we're plotting clusters of communities for each of those land use or intervention types. The closer these clusters are to each other, the more similar those communities are. And we expect that with time, if our intervention is working well and increasing tree cover in our cocoa fields or restoring degraded pasture lands, should be supporting species that are relying on the adjacent forests and making those connections happen, we should see that those communities start shifting to look more and more like the intact forest. So here we have a way to measure, the business as usual. We expect to see something in one quadrant, forest in another quadrant. If we look at the changes in community composition, all of the species that we can identify with DNA, in these two types of interventions, we anticipate that they start moving closer and closer to intact forests. So in the pilot example, we did see this, which was really great news. We're in year 2 of monitoring the first deal for the Amazon Biodiversity Fund, so we don't have great data to show you other than an illustrative example. And we just added 3 new deals that we're monitoring this year as well. So we'll be collecting data on this. And then as we get new data, we can also start modeling how the species richness at that whole farm or across the whole, some of the deals are like farm consortiums. So in that whole region, how is species richness changing over time with these different land uses and forest cover trends? So in summary, it's a pretty exciting approach, I think. And I like that it's a way to start getting information back into land managers' hands to help them understand, I'm trying to do good on the land, I'm trying to support conservation, I'm trying to make a living. Is my intervention working? And also, if it is, I'm sending this information back to, through the MRV process to the fund, and I'm getting payments for this or, low interest loans. And then also the investment the fund managers are also getting more information on where should I be investing my money in to make the biggest impact. So we're really excited to keep collecting these soil samples and running the habitat assessments, and hopefully at another Geo for Good, I can share some news with you about the impact of the Amazon Biodiversity Fund. But I'm excited to share the methods with you.
Thank you so much, Karis. Next up, we've got Charles Gibbons from Nature Metrics.
Hello, everybody. Thank you so much for having me here. It's a great pleasure. I'm Charles. I'm a limey. So what we do is, you come to a conference, a bit like London buses, you wait for ages for one, and then suddenly 2 eDNA providers turn up at the same time. So we use eDNA capabilities to detect the presence of species. Now, if you're going to ask what's more important, camera trapping, bioacoustics, or eDNA, or iNaturalist, or GBIF data, they are all critical. We just do not have enough available data sets at the moment to be able to do, very in-depth restoration. So collaboratively, we have to work, and it's a data sharing and a data platform that we need as a capability that bring things together. So the new regulatory maybe standards coming after Kunming Montreal and launch of TNFD type standards, they are going to be very, very data-driven decisions or recommendations engines that we need to be able to make. So we are a UK and Canada-based company. We've been going about 7 years. We've had about 500 plus projects. There's about 140 of us now, lots of PhDs. And I want to talk to you specifically today about a research project we've been running in Zimbabwe. So in Zimbabwe, between Bulawayo and Harare, basically straight down the middle of Zim, is a 500-mile long mountain range called the Great Dyke. Totally visible from space, and you can see it. Now, what makes the Great Dyke so mineralogically important, or is the mineralogy that makes it important, is that it has a lot of the world's rare earth metals. So palladium, cobalt, gold, everything down to asbestos are available and mineable at this site. So there are lots and lots of individual mines going on at this location. So one of our customers who we're working with here were mining and wanted to know their impact. What was the impact of their mining work in this environment? How did it compare to the relevant other mines? So to be able to do that, they needed to be able to monitor within a mile, a 2-mile, and a sort of 10-mile perimeter of an area, and then use that information, all that eDNA that had been captured, so the thousands of species that were captured from that information, to then make a recommendation. And that recommendation took the form of a beta diversity model, an inverted alpha diversity model. So in the Zimbabwe Great Dyke, we discovered 1,200 different observed taxonomic units. So the eDNA tool, it picks up, comes back to our labs in the UK. We run the meta barcoding process through that. That generates all of the data of the discovered species. We then match that against available data sets that provide us with the DNA sequences. We match the DNA, pattern matching of the base acids of the DNA. That gives us a list of the known species. We then plot those at sites, and then we start building and training a model on joint species distribution modeling. So I've seen a lot of talks. There was a very good talk about EE classifiers earlier in the conference. And there's a single species distribution model called maximum entropy, which is a brilliant model if you want to nominate a keystone species and look at an environment. We wanted to go a little bit further and build a hierarchy or a structure of species of the various taxa, understand their relationships, be able to pass in other information on the dynamic world label, the eastness, northness of, or the slope of and the height and the temperature of where the sample was detected on the Great Dyke. So this map briefly shows the species richness as being the diameter of the circles of all the sampling points in and around the various mine sites that we were looking at. So that goes back into the SJSDM machine learning model. So into that SJSDM model goes the dynamic world labels, the various other attributes, and the graph data structure that we try and build of the taxa that have been discovered. So this is where actually the more data we have, and in the environments that we have, from bioacoustics or from camera traps, and from more and more eDNA monitoring, and from citizen science as well, means we can build a much stronger, the community can build a much stronger taxa and dependency index and model between species in an area. So taking away the points of the species detections, we came up with this sort of visualization of beta site irreplaceability, where the green areas are the areas of criticality. So in areas for able restoration, there were a few areas that we found that had great opportunity for still protection or restoration. Meaning that the mine sites had been quite considerable from all the different types of mining that had been going on in this environment for a very long time, that we needed to then focus very much on the conjoining areas that are shown in the highlighted points in green. So this is what we do, the recommendation engine. We then come back to the end customer, and then we bring in the expert consultancies who then build on a restoration project and then try and restore the land in that environment. So thank you very much for having me. And if you want to know more about eDNA, there's at least 2 of us, so come and see us while you're here.
Thank you so much, Charles. Now we get to hear from Catherine Mertes from the Smithsonian Conservation Biology Institute. Did I get that right? Smithsonian Institute.
There's a zoo in there too.
Yeah.
Thank you so much.
Thank you.
And thank you everyone for the opportunity to share some stories today about how the Smithsonian's National Zoo and Conservation Biology Institute is using tools like Google Earth Engine to protect populations, species, and the landscapes where they live. So today I'm going to talk mostly about 2 species of antelope that live in some of the most remote regions on Earth and some of the harshest environments on the planet, and that are at very real risk of extinction. On the left are several scimitar horned oryx, a large antelope native to arid seasonal grasslands in North, West, and Central Africa. This is the last known image of wild scimitar horned oryx before the species was classified as extinct in the wild in 1999. On the right is a more recent image of Addax, another large arid land antelope, roaming the Reserve Naturelle Nationale de Pintea last year, one of the very few times an aerial or ground survey has located living Addax in recent years. Pintea is home to the last known wild population of Addax, which is only estimated to contain about 50 animals. Oryx and Addax underwent dramatic declines along very similar timelines and due to very similar sets of reasons, primarily increased hunting pressure due to widespread availability of 4x4 vehicles and modern weapons across North Africa after World War II, human population growth and expansion into arid landscapes, primarily for livestock grazing, and ongoing persistent conflict in range states. But almost since the moment that scimitar horned oryx were declared extinct in the wild, conservation organizations were working to try and find partners and places to reintroduce it and similar species. And here we have some historic range information and some potential reintroduction sites that I'll get into a little bit in more detail. And on the right, we have a set of figures from stakeholder workshops showing that multiple releases of animals over consecutive years to achieve a population of at least 500 breeding animals was deemed the scenario most likely to produce viable populations of oryx and addax. And in 2013, the Environment Agency Abu Dhabi partnered with the government of Chad and the nonprofit conservation organization Sahara Conservation to reintroduce both of these species into the reserve de faune de Wadirime Wadi Hashim, a large protected area in central Chad. So large, it's about the size of Maine, about 90,000 square kilometers. There are many collaborators on this reintroduction program that have achieved some truly amazing results, including releasing more than 280 animals into this reserve, or more than 280 oryx, more than 90 addax. And nearly all of these individuals who are released were fit with GPS satellite collars, creating a data set of more than 5 million collar locations and more than 20,000 direct field observations since 2016. So I'm going to quickly cover 3 topics where we're leveraging the strengths of Google Earth Engine with our movement and observation data sets to ensure that these populations reach long-term viability. So first, because these 2 species have been very rare, if not completely extinct in the wild since around the 1980s, most of our understanding of their ecology comes from captive collections. And it's very difficult to know whether findings on, say, birth seasonality in captivity, where resources are abundant and very different than the arid Sahel, are even relevant for wild populations, or what natural social groups are for these species. When in captivity, they're managed to maximize genetic diversity among offspring and to minimize conflict among individuals. And we're particularly interested in learning about these species' natural habitat preferences, because these will determine where reintroduced oryx and addax travel and use resources across the reserve after release. So we combined our collar data in a type of model called an integrated step selection analysis, and we used the massive data catalog in Earth Engine to extract environmental variables that were relevant for these 2 species, closest in time and space to our collar locations used and available, and nearby locations where animals could have realistically visited but did not, and also at a spatial resolution that's appropriate for the movement capacity of these species. And we found some really interesting differences among these species. So addax appear to track short-term vegetation productivity, which we represented as short-term change in NDVI, much more closely, which kind of tracks with our understanding of them historically as a species of the true desert. And scimitar horned oryx appear to have a broad strategy of tracking long-term vegetation productivity and maximizing energy gain during the wet season and minimizing energy loss by avoiding complex topography and long searches for resources during the dry season. We're also using more classic remote sensing analyses to identify suitable habitat into which these populations might expand over time and which might therefore need enhanced protection. And we're also identifying potential hotspots for human-wildlife conflict. However, remote arid landscapes offer many challenges to standard remote sensing approaches, especially fine spatial heterogeneity, strong seasonality, high interannual variation, and buildings made of natural materials that are very difficult to tell from exposed soils and sand. One particular concern in this system is reliably separating grasslands from agriculture. Rain only falls here from about mid-July through mid-September, and all agriculture is rain-fed, so both managed and natural herbaceous vegetation are responding to the same very compressed wet season. And agricultural practices vary so much among different tribes and families that there's no consistent harvest timing that we could otherwise use as a signal to tell apart agriculture from natural grasslands. So to improve separation of these land cover types and an ongoing land cover classification, we chained together a couple user-published repos in Earth Engine. First, to calculate biophysical variables, leaf area index, and fraction of absorbed photosynthetically active radiation for 8-day Sentinel-2 composites, and then to calculate time series and land surface phenology metrics from these composite time series. So I just wanted to shout out these 2 awesome repos and their related papers and highlight that this type of analysis at this spatial region and at this spatial extent just wouldn't be possible for our project in terms of time and cost constraints without Earth Engine. So biophysical variables like LAI and FPAR are much more sensitive to changes in vegetation density and photosynthetic activity than band ratio indices like NDVI and SAVI, and they really were better at capturing the subtle changes in annual grasslands and agricultural crops throughout the growing season. So using this LAI time series, even though harvesting timelines differ among tribes and families, the LSP metrics, length of season and end of season, were still different enough from natural grasslands that they led to really meaningful improvements in our classification accuracy. And we're using this classification to identify enhanced conservation zones where animals are already moving through the reserve and where they may expand into in the future due to the location of abundant suitable habitat. And then the final example I'm going to share is about disease ecology. So sadly, in 2018, about 40 reintroduced oryx died, eventually attributed to a mosquito-borne disease called Rift Valley fever. And we wanted to quantify the changes in movement capacity and habitat selection in infected animals to potentially build some movement-based alerts and other tools to help us detect and possibly prevent massive mortality events in the future. And I wanted to highlight here that in this system, a really important resource is shade. It's used by many species, our reintroduced antelope, livestock, other wildlife, and also disease vectors like ticks. So we used planet monthly mosaics available in Earth Engine, calculated NDVI from them, and summed that across the early dry season and late wet season periods when mostly shrubs are the only vegetation with leaves in this system. And then we then summed across multiple years to create a probability of woody cover layer, which we then validated using tree count and cover estimates from the field. When we combined this woody cover layer with movement data from oryx infected and not infected with Rift Valley fever, we found that infected animals selected woody cover much more strongly, which put them at greater risk of further disease exposure. And like the other examples I showed, this analysis, particularly with this high resolution imagery across such large areas, simply wouldn't have been possible for our project without Earth Engine. So I just have to mention the many people and organizations that are collaborating to reintroduce and monitor these populations in the reserve, and throw up my contact info and SCBI's website if you want to get in touch and talk more about applied conservation and spatial ecology. Thank you.
Great, thank you so much. I think we have about, we have a couple minutes for any questions from the audience. Feel free to step up to the mic. Yeah, excellent.
Thank you so much for the wonderful, wonderful application presentations. Question for the eDNA gang and a question combined for Catherine. So let's say that the antelopes and the animals that you're tracking are roaming the earth, and then do they have any traces of DNA in the soil that you could probably detect from your eDNA samples?
Yes. So these are the little units that we use to collect DNA. We all shed our skin and every other, part everywhere. So, yeah, there's always DNA to be found.
So there you go. That's one monitoring, for where they have been. So on the same track, I used to do microbiome sequencing and 454 sequencing back in the microbiome days of humans. Not one of the heavy criticism labeled just against all of the research in the microbiome region is, well, these organisms might be dead. They are no longer alive. They're just there. It's just, sequences of dead animals. So, but bacteria. So how is that criticism just leveled against your work as well? And how do you kind of tackle that?
That's a great question. I'll say in the area we're working, our problem is preserving DNA because things process really fast. There is going to be some relic DNA in there. I also think that's interesting, kind of getting that history. And if you think about, the fact that you can have some contamination in your soils, how do you incorporate that noise into the studies you're doing to learn from it? So I think like in the case of what we're doing, we're looking for changes. So we're kind of accounting for that legacy. But it's a concern, and I think it's a concern depending on, more or less of a concern depending on what your question is. And then just a quick cool side note, butterfly wings, I guess, are like, stay preserved in the soil for a long time. I'm not a molecular biologist at all. I was just reading some of this. And they have an interesting study where they're recompiling like the history of some of the butterfly ecology from butterfly wing data in the soil. And I can share the paper with you if you want. The paper will have much more detail than I can provide. I don't know if you want to add to it.
Thank you.
Hello, thank you for your presentations. It was really interesting. I was curious if you could shed some light on finding the balance between making your data of all these species open to the public to support open science and validating data with other data sets from different sources versus keeping the endangered species data protected in the sense of, making sure that poachers don't pursue the locations that you're exposing these animals to be in.
Maybe I'll take a step. Yeah, we face that problem constantly. And I think that my natural inclination is that this is information that is important for conservation. On the other hand, there are concerns about making the data identifiable in space or time. And so we've come up, at Wildlife Insights, we've come up with ways to obscure the locations of sensitive species so that people can still do a lot of work with them without knowing exactly where the species are, in the region where they are, and that strikes kind of a healthy balance where you can still share some of the information, but not all the detail of the information. And of course, there's always going to be people that would like to have access to those locations, but then those people can get in touch with the data users that collected the data and then share the data privately rather than publicly. But I think there's an important, as long as we have safeguards that do not conflict with the conservation goals that we're trying to achieve with species, I think it's good to have open data. And again, there are some places where data must remain private because of other concerns, and many indigenous communities, for example, are hesitant to share data, and then that has to be respected. And as long as that data can be used at an aggregated level, I think it kind of helps achieve some outcomes on how to use it.
Great, well, we're up on time. Thank you so much for coming, and thank you so much to all of our speakers for sharing all your knowledge and wisdom with us. Round of applause.